Hello everyone, welcome to BioScholars Academy of Learning. I'm very happy to meet you all with yet another video of LAC OPERON. So in the last class or in the last video we were discussing about the basics of LAC OPERON, and we shall continue with the concept today. Okay. So LAC operon is an inducible operon. So what do you understand by inducible operon? So inducible operon is a very simple concept. It is induced, or it is on in presence of a particular inducer. Okay. So an inducible operon is one that is expressed in the presence of a substance termed as inducer. Okay. So now let's understand what is this inducer. An inducer is a small molecule that triggers the gene transcription by binding to the regulatory protein. So we saw in the last video that the LAC operon is basically regulated by a repressor protein or a regulatory protein, right? So this inducer basically goes and binds to the repressor protein and thereby regulates the transcription of all the LAC genes. Okay. In LAC operon, lactose and its analogs are termed as inducers. Okay. So actually, a chemical called allolactose. Lactose is basically converted into allolactose. Allolactose is the actual inducer of the operon. Okay, and analogs. Analogs means these are chemically made or synthetically similar compounds which are similar to allolactose. Okay, they act as inducers, like your IPTG. It is a synthetic inducer of LAC operon. Isopropyl thiogalactosidase. Okay, fine. Next slide. LAC repressor. So we already understood that LAC repressor is a product of the regulatory gene that regulates the transcription of the structural gene. Okay, so a gene that codes for a protein involved in regulating the expression of the other gene is the regulatory gene. So it's called the I gene. Okay, so I gene can block the expression of LAC, Z, Y and A. So we saw in the last class how the last video how this repressor gene is going to go and bind to the repressor gene produces a repressor protein, and how this protein goes and binds to the operator and thereby blocking the expression of the LAC, Z, Y and A structural genes. Okay, so let's try to understand what is a LAC repressor. LAC repressor is a homotetramer, meaning it is made up of 4 compounds which are exactly similar, okay, with a molecular weight of 38,000 Dalton. And repressor binds to the major groove of the DNA in the operator region. So I earlier told you repressor goes and binds to the operator region. So in DNA, you know, you have two things. One is your major groove and other one is your minor groove, right? So your repressor protein goes and binds to the major groove of the DNA, where in the operator region. Okay. So this what is this repressor doing? It is going to go, if this is the LAC operon gene, if this is the operator, okay, and this repressor will go and bind to the operator region. Okay, and what happens, there is by binding to the operator, the repressor prevents transcription initiation by RNA polymerase. So if you all remember, next to the operator you have a site called promoter, and you know RNA polymerase goes and binds to the promoter region, right? So as a result of as a result of your repressor protein binding to the operator, your RNA polymerase cannot move any further. Okay, and this is how the repressor protein is basically working, if you understand. Okay, so LAC repressor molecule has 2 recognition site. Okay, so one is the DNA binding site that can recognize the specific operator. Okay, so when does this happen? In absence of lactose, or when there is no inducer. When there is no inducer, what happens? The repressor will go and bind to the operator. When there is a inducer, okay, when there is an inducer, what happens? Say for example, if this is the repressor protein, your inducer will come and bind to the repressor protein. Okay, it changes the configuration of the repressor protein such that the repressor protein can no longer go and bind to the operator, and as a result you can see all the structural genes will be expressed. Okay, yes, I think I have a picture. So here you can see this is the regulator gene. Okay, so when this regulator gene, it is going to produce, one second, it is going to produce a regulatory protein, and this regulatory protein goes and binds to the operator. Okay, goes and binds to the operator. Fine. So now what happens if lactose is present in the medium? The lactose will bind to the regulatory protein, otherwise called the repressor protein, and hence the repressor protein cannot go and bind to the operator, and hence the structural genes will be transcribed very successfully. Okay, this is what is happening. Okay, in short, when there is no inducer, that is the lactose, the repressor binds to the operator, thereby inhibiting the transcription of structural gene. It's exactly what I said now. Okay, when there is inducer, the lactose, that is your lactose. What is the inducer of the LAC operon? Lactose. Okay, it's actually allolactose, but in colloquial we just tell them as lactose. Okay, allolactose and lactose are isomers. Okay, the repressor binds to lactose, which changes the conformation of the repressor, thereby repressor cannot bind to the operator. The same thing I told you, right? So now transcription takes place, resulting in the synthesis of structural gene, which helps in the metabolism of lactose. So in the last class we studied, so LAC produces beta galactosidase, right? So beta galactosidase helps in conversion of lactose into glucose and galactose. Okay. Similarly, permease helps in taking up of the lactose into the cell. Okay, and transacetylase helps in the transacetylation reaction of the molecules. Okay, so these are the three structural genes. Okay, now we have to understand the negative regulation of LAC operon. Okay, so what is negative regulation? Whenever there is no inducer, the operon should be switched off, right? That is called negatively regulated. Okay, how is it negatively regulated? Because the LAC repressor, so here you can see the LAC repressor goes and binds to the operator. Okay, and prevents the transcription of the LAC operon. This is the overall understanding. Okay, so what happens, because the binding of the repressor prevents transcription, we call this LAC operon as a negatively regulated operon. Okay, so very simple, when there is no lactose, the operon is switched off, the operon is off. Okay, and this is called as negative regulation, as simple as it can be. Okay, the LAC repressor molecule is a negatively regulated and acts as a roadblock on the DNA. So say for example, if this is your LAC operon, okay, again and again I am drawing it so that it sticks to your brain. So you will see your repressor going and binding to the operator. Okay, so it is like a roadblock on the DNA so that the RNA polymerase cannot move freely. Okay, as a result, the structural genes of the LAC operon are repressed. Okay, it acts like switching off the LAC genes. Okay, yes, so here very clearly they have given, you can see the repressor. Okay, whatever I have explained, the repressor is a homotetramer. As I already said you, it is having 4 similar units or 4 monomers, and here you can see RNA polymerase is bound to the promoter site. Always remember this, RNA polymerase binds to the promoter site along with the sigma factor. Okay, then you can see the repressor protein is getting bound to the operator region. And one more thing, why it is active? Because there is no lactose in the medium, and as a result the repressor remains active. Okay, and hence the transcription is blocked. Okay, now in presence of lactose, what is happening? The lactose is converted into allolactose, and allolactose binds to the allosteric region of the repressor protein. Okay, the allolactose binds to the allosteric region, and now what is happening is, you can see the operator is free from the repressor protein and all the genes are transcribed. The operon is on. Okay, so in absence of lactose, the operon is off. In presence of lactose, the operon is on. So this is the concept which we all need to understand. Okay, when the inducer is present, it binds to the allosteric site of the LAC repressor. Okay, this site is called as the allosteric site, regulatory site. In other words, we can call it as a regulatory site. Okay, however, it's not lactose which is an inducer, but it is allolactose. Okay, so I already told you what is allolactose, right? They're isomers. Okay, lactose and allolactose basically differ in their linkage. So they differ in the linkage between glucose and galactose. Okay, so allolactose is an actual inducer of the operon. Formation of allolactose is catalyzed by beta galactosidase. So beta galactosidase has 2 functions, right? One is breaking off lactose into glucose and galactose. Another one is conversion of lactose into allolactose. So you can have a question, which enzyme converts lactose into allolactose, and also which enzyme breaks down lactose into glucose and galactose. Okay, so these are the two possible questions. Okay, next we are going to discuss about Cretaceous inducer. Again, this is a very important terminology. Okay, it is a lactose analog. Okay, and it is synthetically produced. Analog means a substitute. Okay, or substitute which is very similar. Okay, that is capable of inducing the lac operon while itself not serving as a substrate for beta galactosidase. Okay, it induces the operon. Okay, but it is not the substrate for beta galactosidase. Try to understand this difference. Okay, it is going to induce the operon, but it is not going to act as a substrate for beta galactosidase. Okay, so you have a particular chemical called IPTG. Okay, IPTG is isopropyl thiogalactosidase. It's a synthetic non-metabolizable analog of the lactose. Okay, it is going to induce the operon. Okay, but this is not the substrate for beta galactosidase. Okay, but what is a substrate? X-gal. Okay, X-gal is a substrate. So you should have learned this in blue-white selection, right? Blue-white selection. I will do blue-white selection in one of my videos one day. But for now, try to understand what is a Cretaceous inducer. So a Cretaceous inducer is something that is going to induce the operon, but the inducer is not going to act as a substrate. Say for example IPTG. So IPTG is the inducer, but the substrate here will be X-gal. Okay, so I hope you understood all these terminologies. We will start with LAC mutations in my next lecture video. Don't forget to like, share and subscribe to my channel. If you have any queries, please post it in the comment section. If you want me to make videos on any specific topics, any learning strategies, you can just put it in the comment section. I'll try my level best to make them. Thank you so much. Stay tuned.